Dr. Samarani, the Associate Director for Science at the FDA, and he will be talking to us about IVPT fundamentals, scientific and practical considerations.
Hello, my name is Sam Rani, and thank you for that very kind introduction. I've spent years, decades in academia, industry, and the FDA. And through that time, I've developed and validated hundreds of IVRT and IVPT methods. So the point here is that I've been a student of the skin and of topical and transdermal drug development for many years. I hope today that I can share with you some of the fundamental lessons that I've learned. And I look forward myself to learning a great deal more throughout the course of the next 3 days. We have a fantastic program and an exceptional faculty with practical real-world experience in the implementation and data analysis. And I think we'll all really have something very special to look forward to. So I'll begin with a disclaimer that this presentation reflects my views and should not necessarily be construed to represent the FDA's views or policies. Now, when we talk about in vitro permeation tests, that permeation that we're referring to is the permeation through skin, and in this context, really human skin. So we'll take a moment to orient ourselves to the structure of human skin. What we see here is that there's a large region that's really known as the dermis, and above that is the epidermis. The uppermost part of the epidermis is the stratum corneum. And this is really considered the primary permeability barrier for the skin. It prevents us, who are comprised predominantly of water, from desiccating and drying out in our dry environment on land here. It also is a barrier to infection, and it's a barrier to the absorption or permeation of topically applied compounds. So let's focus just for a moment on the differentiation of these cuboidal cells at the base of the epidermis as they differentiate up. So this is often called the stratum basal or the stratum germinativum. And as these cells differentiate upward over a course of several weeks, as your skin replenishes itself, there's a differentiation that happens that creates these flattened plates, essentially, of cells that are corneocytes. And what also happens during this time is that lipids are extruded and ordered in between these corneocytes in a very specific and structured way. And I'll talk about that in just a moment. But one of the things to understand here is that this layer, the stratum corneum, is a non-living layer like hair or like fingernails. And so the barrier properties of the stratum corneum are maintained even after excision from the living body. Here you see a side view of the structural organization of the stratum corneum with the corneocytes here. And you can see what has been often described as a brick and mortar type structure. Over here, this is the top view of those flattened corneocytes. And the classical concept of diffusion of topical compounds is that there's this tortuous diffusion pathway through the intercorneocyte lipid space, through that mortar, if you'll have it, and that the barrier properties are very much associated with these well-ordered, highly structured lamellar lipids, these sheets of lipids that fill in this intercorneocyte space. Now, compounds or even excipients can disrupt this ordering. They can alter the solubility or the diffusion through these lamellar lipids. And that diffusion that we're going to talk about is flux, the flux through the skin. Now, Katz and Poulsen adapted Fick's law of diffusion, and I won't have too many equations here, but I want to highlight really the fact that this flux is dependent upon a few key principal elements. One is the partitioning of the compound from the vehicle, from the dosage form that's on the skin, that partitioning into the stratum corneum, and then diffusion through those intercellular lipids, as well as the concentration gradient, where you have a high concentration gradient at the top of the stratum corneum, and then as it permeates through the stratum corneum, there are mechanisms by which the dermal capillaries clear drug and maintain a concentration gradient. And this has implications for the way that we design IVPT studies as well. Now, in reality, it is a little bit more complicated, as you can see in this equation, which is the finite dose equation. And the key message I want to get across here is not to go through the equation, but rather to say that this is relevant to clinically applied thin film doses. That when we're talking about what happens when someone uses a topical product, they apply it as a very thin film, they shear it onto their skin, and often it changes. It changes across the duration from the time that it's dispensed from the product container as it's being applied on the skin, and then as it continues to undergo metamorphosis where after the rubbing and the shearing, there's evaporation of volatile components, there's a change in the concentration of the drug in that residual formulation, changes in thermodynamic activity, the dose becomes depleted from the dosage form as it permeates into the skin. And so this equation accounts for many of those factors, not everything, but the key is that fundamental scientific principles of an IVPT study are anchored to the clinically relevant parameters of the finite dose model. So let's talk a little bit about the formulation qualities of products that are applied on the skin. Once they're dosed, most formulations dry. So the stability and the solubility change. And it's only that soluble molecular form of the drug that can diffuse through the vehicle and then partition into the skin. The partitioning of the drug from a multiphasic dosage form, like an emulsion into the stratum corneum, can be different depending upon the phase in the formulation from which the drug is partitioning. So, for example, if the drug is inside a globule, and that globule, if you kind of imagine a cross-section where the globule is impacted right down in contact with the stratum corneum, it might be partitioning directly from the globule into the stratum corneum, or it may be going through the continuous phase and partitioning from the continuous phase into the stratum corneum. That partitioning would be different. Also, as formulations undergo metamorphosis, the thermodynamic activity of the drug in the dosage form changes. So as the drug becomes more concentrated in that dosage form, its thermodynamic activity can increase. And this change can modulate the rate and extent of cutaneous bioavailability. Also, the arrangement of matter in the dosage form can modulate its structural interface to the skin, its metamorphosis, and the rate and extent of cutaneous bioavailability. Now, the excipients themselves also penetrate skin, and they can alter the solubility of the drug in those intercellular lipids of the stratum corneum. They can alter the ordered structure of those intercellular lipids and even create domain phase separations and affect the penetration and or perhaps even enhance the penetration through a variety of mechanisms. The point is that excipients can impact the partitioning and the diffusion of the drug. So in context of the in vitro permeation test, this is essentially a simplified diagram of a diffusion cell. This is a vertical diffusion cell, often called a static diffusion cell. Static because the receptor solution stays in there between samples. And what's focal point here is really the membrane. The membrane in this case is the skin. So many times the skin may be dermatomed, that means horizontally sectioned, and many times sectioned at about the level that the dermal capillaries would clear permeating drug from the skin and maintain that concentration gradient, except here, the receptor solution is the one that receives that drug and clears and solubilizes it and maintains that concentration gradient, which is very important. There are times when there are epidermal preparations where the epidermis may be separated and the epidermis may be mounted. But the general concept, the most important thing, is that the skin must be maintained at physiologically relevant conditions. So that's physiologically relevant hydration and temperature. And so this diffusion cell is designed to do that. The receptor solution is a physiological buffer. It has a water jacket around it in this implementation to maintain the temperature of the system such that the skin surface temperature is 32 plus or minus 1 degrees conventionally. And then it maintains the surface of the skin open and dry to the external environment, just like it is on our bodies. Excuse me. Another implementation is a flow-through cell. The design is relatively similar, where the skin is mounted in the diffusion cell The compound is applied on top, it permeates through just as it does here. But here the receiving fluid, the receptor fluid, continues to flow through, and we'll talk a lot about the differences in these designs, both of which can create very good IVPT studies. IVPT key features are summarized on this slide. So the membrane is typically excised human skin. The receptor solution or the medium is typically a physiological buffer. The methodology maintains the skin at physiological conditions. The dose is typically a thin film for non-steady state kinetics, because the dosage form is changing on the skin continually. Typically, the dose is not occluded to allow for that drying or metamorphosis, and the primary result is the dynamic rate and extent of permeation of the compound of interest through the skin. Now, this permeation may be very small because the drug is often released into the receptor solution in the nanogram per mil or picogram per mil range even. The dosage form is maintained on the dry surface of the skin, so it doesn't come into contact with the receptor solution. And validated IVPT methods can exhibit very similar flux profiles within a donor. So within a donor, you can really make comparisons and have comparable flux profiles. IVPT results, as we'll talk about, can correlate with in vivo results when there are harmonized in vitro and in vivo study designs. And this rate and extent of permeation can be quite sensitive to changes in the physical, chemical, and structural or Q3 attributes of the skin. Now, here is some examples of an IVPT, of results from an IVPT study, in this case showing the flux being the rate comparing different formulations. And the metamorphosis of these formulations is different, and that's what creates these different flux profiles. Here, flux is a rate, so that's like saying, 10 miles per hour, 20 miles per hour, 30 miles per hour. This is a constantly changing rate. It's dynamic. It's not steady state kinetics. And that's important to understand, that IVPT is clinically relevant and measures the non-steady state rate of skin permeation. And Dana, I wonder if someone might have their microphone live right now, because I'm hearing a little bit of feedback. This is by contrast to an in vitro release test, which has steady state kinetics and measures the steady state of drug release, not permeation, but it's one steady state rate. Now, the other thing I want to talk about is what is the parameters. We looked at flux data. I also want to talk about the other parameter that we often look at, which is the cumulative amount permeated. And this is a plot showing not flux, but the additive amount that's permeated across time, the cumulative amount permeated. This has historically been called AUC for the area under the curve of the incremental permeation profile, but it is different than the AUC used in, let's say, plasma pharmacokinetics, where there are calculations with trapezoidal rules. So here, I'm proposing that we use a distinct term, not AUC, but AMT for the cumulative amount, where that amount is really this value, the additive amount across all the samples that's permeated across time. That cumulative amount is that AMT. And this can be kind of contrasted with the flux.
Yeah.
You see,
I got an order to pick up. Yeah.
I wonder, Dana, if you could mute some of the other participants at the moment. I'm so sorry for that. The cumulative amount permeated is shown here, and this is the same data for flux. And you see that the flux can be a little bit more differentiating. In this case, we're looking at data that happens to be differences in dosing for a positive displacement pipette compared to an inverted HPLC vial used to spread it on the skin. And you can see that the difference in the way that the dosage form undergoes metamorphosis, even during administration, can affect the rate and extent of bioavailability. There's other data for this as well, showing the influence of dose dispensing, just even dispensing from the product container, on the quality of the product. Here we see as acyclovir cream for a product in Europe that's marketed both in a tube and in a pump. And when you look at the product inside the tube, or if you open up the pump and look at it inside the pump, you don't see dimethicone globules, but after the product in the pump has been extruded through the shear forces of the pumping through the nozzle, you can see that the dimethicone, which is blue in this Raman image, dimethicone comes out of solution and these dimethicone globules are evident. This is also shown in a 3D reconstruction of the Raman data. You can really see those blue dimethicone globules. What we see here is that the performance of this product is different when it comes out and the structural arrangement of matter in the dosage form changes. So this is IVPT results from multiple labs. These are part of FDA collaborations. And what I'd like to highlight here is that although the different labs in different parts of the world used different diffusion cells, different skin preparations, different dosing techniques, and other conditions being different, what we see here is a very consistent outcome in the results where for the reference product, which was the US reference product in green here, when that was compared to a negative control for bioequivalence, this product in red, it was consistently differentiated in all the studies under all the conditions. Now, the other thing that we see here is that the product in the tube, the UK product and the US product, were found to be comparable independently in the different labs, and the product in the pump was found to be differentiated from both the negative control and positive control in both the studies. So what we see is that in these completely independent studies, when the test and the reference products are compared under consistent IVPT conditions within that lab, using a well-controlled method that really accounts for the scientific principles of the test, that you consistently get meaningful information about the relative rate and extent of the compound from the test and referent products. So how does this relate to in vivo data? Well, there's been a lot of historical information about the in vitro, in vivo correlation, in this case for scopolamine, where it was reported that the in vitro accurately predicted the situation in vivo. Also for benzoic acid and petrolatum, the black data here is the in vitro rate of absorption and the in vivo rate of excretion. Now, this is rapidly converted to hippuric acid and then excreted. This is some data for testosterone in vitro and in vivo, as well as estradiol in vitro and in vivo. And here we see on the left side, halobetazole propionate, and on the right side, alclometazone dipropionate. What we see on top here, these open shapes are the ointments of each. And at the bottom, these filled shapes are the cream of each corticosteroid. And in each set, this is comparing test versus reference. The circle versus test versus reference in each set. So these were test products that were designed to match the rate and extent of the reference product. And as we can see here in the IVPT study, that the IVPT study said they matched quite well. When you look at these same products then evaluated in vivo, these were corticosteroids, so they were evaluated in vivo using a vasoconstrictor assay. And with one exception, these were not only the same product, they were even the same lot of the test and reference products. They were evaluated using a vasoconstrictor in vivo clinical assay per the FDA guidance and found to be bioequivalent. The test to reference ratios are very close to 1 in both instances. And this is the data to which Dr. Maybach was referring previously in his keynote address, where one of the challenges for many years were that there were conflicting reports in the literature about the usefulness of IVPT. Some researchers reported excellent in vitro and vivo correlations, some did not. And this work was very helpful as a retrospective analysis to deconvolute this discrepancy in the literature. It compared 92 IV, that's in vitro and in vivo data sets. And what we see here is this black line is the line of perfect one-to-one correlation. The dotted lines are plus or minus threefold differences. And then we go beyond threefold differences. And because of the large data set and the range of permeation, this is on a log scale. While there is a kind of correlation that you can see here, I think it's very important to really point out that the in vitro in vivo ratios ranged from about 0.2 to roughly 20, meaning that you could have a 20-fold difference in the amount reported to be absorbed in vitro versus in vivo. That's pretty substantial. Now, one of the issues with the data set is that these were not intentionally designed to be compared in vitro and in vivo. And in fact, sometimes they had different formulations that were dosed in vitro and in vivo on different anatomical sites. And there was regional variation in the permeability of human skin. So if you dose it on a different anatomical site, you're likely to have a different permeability. Now, there was a subset of these studies, 11 of these, where the formulation that was dosed in vitro and in vivo and the anatomical site used were well matched. And when you look at just that subset where the data study designs were harmonized, the correlation is now much closer to that ideal line. And also the range for the in vivo to in vitro or in vitro to in vivo ratios now was 0.58 to 1.28. So not even a twofold difference in this case. And the overall mean was about 0.96. So one of the take-home messages here is that when you change the conditions, and this is whether you do it in vitro or in vivo, if you change the conditions, use a different formulation, a different dose duration, different method for administration or rubbing into the skin, a different anatomical site for skin, or even use skin from different individuals, that you should expect that there may be differences in the rate and extent of bioavailability. Now, the corollary to this is that you should always compare under harmonized conditions, whether you're comparing test versus reference or in vitro to in vivo. Now, this was a retrospective assessment done on existing studies in the literature. This lesson learned was then applied to studies prospectively going forward, and I'll show a little bit of that data. So here we have lidocaine prilocaine cream. It's a 2.5% lidocaine, 5% prilocaine. And what we see here is that we compared the reference cream and the generic cream. So these are products that have already been clinically demonstrated to be bioequivalent. So they're a positive control for bioequivalence. And as a negative control, there was a gel. They have different physicochemical and structural properties, but what I really want to get across is we know, based upon clinical evidence, this is a bioequivalent product. And these positive controls for bioequivalence, when compared using an IVPT, showed a comparable rate and extent of cutaneous pharmacokinetics of bioavailability. And when compared to the gel as a different formulation, as a negative control for bioequivalence, was nicely differentiated. So we see here that the IVPT study corresponded well with clinical in vivo bioequivalence data, and that when you look at harmonizing the study conditions and now looking at the cutaneous pharmacokinetics in vivo, we see here the same products, well, certainly the reference cream, and this is in vivo DOFM data where probes were inserted in the skin, and the same negative control gel were nicely differentiated where the cream again exhibited a higher bioavailability than the gel. So the IVPT results corresponded with clinical in vivo results, not only in terms of the pharmacokinetics, but also in terms of the clinical bioequivalence for this approved generic. Now, this was the data for lidocaine, but there was also prilocaine in the product, and the data is very similar for the prilocaine, where you can see that the positive controls were found to have very comparable pharmacokinetics and differentiated from the negative controls, and we see very similar results for the prilocaine in vivo as well. We have another example here with metronidazole, where again we have the reference product compared to the approved generic, and so these are demonstrated to be clinically bioequivalent. When these products were compared by IVPT, they were found to have very comparable rate and extent of permeation. Likewise, when you look at the metronidazole in the gel formulation compared to its generics as positive controls, we see they have a very comparable rate and extent of permeation. So within each set, the positive controls for bioequivalence were very closely matched and discriminated from each other. In fact, these other data sets, multiple products, multiple drugs, multiple dosage forms, we've consistently seen this, including with acyclovir, not shown in this presentation. But there is one additional one that I'd like to show, and that is for a transdermal product. Most of the time when we talk about topical products, we're comparing that to in vivo results in the skin. And so, the previous data was comparing it to probes, micro perfusion probes inserted into the skin and looking at the local rate and extent in the skin. One of the things with transdermal products is that we can evaluate the plasma pharmacokinetics. It's relatively straightforward to monitor the plasma PK for the systemically acting and systemically absorbed transdermal compounds. And this was some work done where the IVPT study was done under harmonized study designs at the University of Cincinnati and at the University of Maryland. And that same harmonized study design was then done in vivo at a clinical site also at the University of Maryland. And then the in vitro data from both universities was used to predict the in vivo data. What we see here on the left side is the data from prediction from the IVPT studies done at the University of Cincinnati and the IVPT studies done at the University of Maryland. And here what you see is at the top, the heat. There was some heat that was being applied because we wanted to understand the effective heat on the rate and extent to which drugs were delivered from these transdermal systems, in this case nicotine, whether the heat was applied early, as it is in the top panes, or whether it was applied later, as you see in the bottom panes. But it's really quite remarkable here that using a harmonized study design, it showed excellent in vitro, in vivo correlation. But I want to make sure that the right message is taken across from this slide. Although IVPT can show excellent in vitro, in vivo correlation, there are more complex things that can happen with the absorption, distribution, metabolism, and excretion. And we see that very much in transdermal products. In fact, there's additional studies that were done just like this with harmonized studies with other compounds. And it was not so easy to see this very nice overlay of the data because, for example, the in vivo rate of elimination, the clearance rate, was elimination from the skin and clearance through the systemic circulation. The rate of that was quite variable in vivo, and so that made it an order of magnitude difference of variable, and that made that prediction harder to match. And there may be other reasons why. The message here is not that a IVPT study will tell you the precise quantitative amount that you will measure in vivo, but rather it indicates that a well-controlled IVPT study that adheres to the key scientific principles of the test provides clinically relevant information about the relative rate and extent of bioavailability from test and reference products when they're compared under comparable conditions. So what should those conditions be? Well, a typical IVPT study design involves taking skin from a donor, from an individual, we call them a donor, and typically taking multiple replicate skin sections. This is mounted on a diffusion cell and dosed with the product, and then as that sampling, that pharmacokinetic sampling from the diffusion cell gives you that cutaneous pharmacokinetic profile. Now that's done not only with the test product, but with the reference product using skin from the same donor, compared in the same donor using multiple replicates. And then this is repeated with skin from another donor and another and so forth using a population of skin donors. And you have multiple donors and multiple replicates per treatment. And that's essentially a routine, a conventional design for an IVPT bioequivalent studies comparing a test and reference product. Now, what are the parameters, the method development parameters that help you to determine how to develop a suitable IVPT method? Well, often you can either use a vertical diffusion cell or the flow-through cell. You typically want to use a finite dose, for example, something in the range of 10 milligrams per centimeter squared. Could be less, could be more. Can often be standard in terms of the stirring rate. The sampling amount, the receptor sampling, can be adjusted, but as you take larger sample volumes, you may reduce the errors, because keep in mind that you have nanogram to picogram per mil amounts of drug that are in the receptor solution, and that the amount permeating, the change in that amount from one time point to the next may be very, very small. And so if you have aliquots, small aliquots, where the error in your sampling may not be precise enough to be able to pick up the difference, you might get senseless data that suggests negative flux or things that other kind of aberrations, where you get zigzaggy kind of data rather than kind of smooth flux profiles. In terms of the sampling schedule, what you want to think about is a sampling resolution for the flux profile that allows you to well resolve it, and that the receptor solution should have suitable solubility for the drug without altering the skin barrier. So there are some, many topical drugs are hydrophobic. And so to solubilize them in a physiological buffer may require using solubilizers. But if you use organic solvents, those organic solvents can permeate up through the skin and alter the skin barrier. And then that violates the principle of the test. The skin source is typically from cadavers or surgical skin to excise that. The skin type can depend on the anatomical location, could be posterior torso or abdomen or a thigh. The skin preparation can be different. It could be dermatomed, it could be heat-separated epidermis. And you always want to have a barrier integrity test. Often conventionally, there are 3 transepidermal water loss, some electrical-based methods for impedance or conductance, and then radioisotope like tritiated water approaches. The goal here is to make sure that the barrier is intact. Now, in terms of how you know whether you have suitable IVPT performance during method development, a sampling schedule and a study duration should be sufficient to characterize the cutaneous pharmacokinetics of the drug, meaning that the sampling frequency selected is selected to provide a suitable resolution to the slug profile. Typically, this means a minimum of 8 non-zero sampling time points across the study duration, which may be 24, 48, 72 hours. You also want to have a sufficiently complete flux profile to identify that maximum peak flux, and then ideally the decline of the flux thereafter across multiple subsequent time points. You want a dose that can be applied in a consistent manner, that provides reproducible flux profiles, that provides a suitable shape for the flux profile so that you can identify peak flux, and that results in sufficient drug permeating that you can actually quantify it with your analytical method. Now, this can be done with a dose that remains on the skin for the duration of the study and that may undergo sufficient metamorphosis to produce a suitable flux profile. Alternately, there may be ones that, like an ointment that just continues to deliver more and more drug over time, where wiping the product off, where you apply it for, let's say, 4 to 6 hours, wipe it off, and then continue to monitor the cutaneous pharmacokinetics may also be a suitable IVPT method during method development to consider. One of the things I want to note here is that when dosing multiple diffusion cells, one of the things that can happen is, let's say you start dosing at 7 a.m. and it takes you, let's say, 30 seconds. You dose, you move on to the next one, onto the next one. So you dose 60 cells, 2 per minute. So 60 cells, and by 7:30, between 7 and 7:30, you've dosed all 60 cells. But then you go to sample it, and maybe you're doing a complete receptor volume removal and replacement. So it might take you a minute. And what this would mean is that, let's say that you're now doing sampling at 9 a.m. So your first time point for your first cell, went from 7 a.m. to 9 a.m., you're at exactly 2 hours. But because it takes you 1 minute each, by the time you get to your 60th cell, it's now taken you an hour, and you've not gotten good control over the difference in the time point between dosing and sampling from the time that you dosed it to the time that you sample it. So it can be very helpful to stagger it for whatever your rate limiting step. So if it takes you a minute for your rate limiting step, then you might want to say, okay, at 7 o'clock, I'll dose at 7, I'll record the time, and then I won't wait until I'll wait until 7:01 to dose the next one, 7:02, 7:03, and so forth, so that my sampling is 9, 9:01, 9:02, 9:03, and so forth. One of the other things that's really quite valuable to have here is a witness during that dosing procedure to make sure that when the operator is dosing it, that they have the right setting, that the setting has not drifted on their pipette, that they've pulled the right product and dosed it to the right cell, because these are things that can be difficult to reconstruct after the fact, and it's very helpful to make sure that that's well controlled and there's no doubt that the correct product was dosed in the correct amount to the correct diffusion cell. Now, when it comes to the skin barrier integrity test, the goal here is to identify and really exclude skin sections with a compromised barrier integrity. But, if there's a tear or a rip or a hole in the skin, there's something that means it's not intact. Now, this test may not correlate with the permeation of the subsequently applied compound. This test is typically based upon the flux of water through the skin barrier. And so the flux of hydrophobic compounds that you may apply later may be different on a particular donor's skin. It's very important that the technical procedures of the barrier integrity test should not irreversibly alter the skin barrier. So for example, if the test involves hydrating the stratum corneum, for example, a pulse dose, let's say a 5-minute dose of tritiated water on the skin, that sufficient time is afforded for the stratum corneum to return to a normal state of hydration, dryness, before dosing. So for example, if the barrier integrity test involves looking at impedance or conductance, and you've left a solution on the stratum corneum for half an hour or an hour, think about being in a bathtub where your fingers are wrinkling because the stratum corneum is getting so overhydrated and mushy. That's not the condition in which the system is now equilibrated and ready for dosing. Likewise, if the test involves altering the temperature of the skin, then sufficient time should be afforded for the skin surface to return to 32 plus or minus 1 degrees before dosing. In terms of what's an acceptable cutoff, the acceptance criteria for including or excluding or passing or failing the barrier integrity test, this can be done based upon looking at that particular methodology for the barrier integrity test used in a given lab and looking at the distribution of results from multiple donors with multiple replicates, so you can get a sense of what is the normal distribution, what is the normal range in that population, and where do you think there's a cutoff that represents an abnormal water flux as measured by your test. That can then be used to say, well, this is the point at which I think I'm going to use this as a cutoff. Everything below this, I think, is intact and normal skin. The second part is then to validate that by actually intentionally damaging a piece of skin and measuring it before and after the damage and saying, before it passed my barrier integrity test, after it was damaged, it was intentionally and deliberately compromised the barrier, that it did actually fail my test. Then I validated that the conditions of my test and my criteria for acceptance are suitable. Let's move now from method development into method validation. One of the most important things to understand is that by the time you've developed your method, you now know what your receptor solution is, you know what your matrix is, and your sample analytical method should be validated so that you're now using a validated analytical method for the rest of the IVPT validation. So you first validate the analytical method, and now you use that validated method to evaluate things that, for example, to evaluate the receptor solution solubility or stability. And then many of these components are actually evaluated in a pilot study. So after you develop your method, you go ahead and run a study, and I'll talk about that pilot study first. So this is typically performed with 4 to 6 skin donors, and typically performed with at least 4 replicates per donor per treatment. And that treatment includes a test and reference product as well as a third product. So think here about those positive controls for bioequivalence that I showed previously, for example, with the lidocaine pralocaine product, and then compared to that negative control for bioequivalence, a different product that has a differentiated flux profile to show that you can hopefully have your test and reference product in your pilot study show very comparable cutaneous pharmacokinetics and show that they can differentiate that from the pharmacokinetics of another product. This supports a validation of the IVPT method parameters like your permeation profile, the range of sampling, the precision of sampling, the reproducibility and selectivity. And this can be really useful to also estimate, based upon the variability of the data, the number of donors and replicates that you're going to need to power your pivotal IVPT study. I'm just going to go through each of these parameters of the method validation very quickly. The first is qualification of the membrane, the skin. So typically, how is this done? Skin thickness is measured and reported for each skin section to ensure that it's relatively consistent for the test and reference treatments. So the assignment of replicate skin sections from a donor to each treatment group, test or reference, is randomized, and the distribution of skin thicknesses in each treatment group can be balanced. If you're concerned that, well, I randomized it, but now I've got all my test samples of skin are really thick and all my reference ones are really thin, this is not a well-balanced distribution. So if there's a pre-specified protocol specified in the study protocol that can eliminate that bias, then that can be utilized. The barrier integrity of each skin section dosed in the study is qualified by, typically one of three barrier integrity tests: TWL, treated water, or electrical impedance or conductance. And this is what's involved typically with qualifying the skin. The receptor solution qualification involves looking at the composition and pH of the receptor solution and looking really at the stability of the drug in the receptor solution samples. This is validated as part of that receptor sample analytical method validation. Looking also at the solubility of the drug in the receptor solution. So often 0.1% or 0.2% of polyoxyethylene 20-oleal ether, also known as Olith 20 or Volpo 20 or Bridge 20. This is commonly included because this little amount in a physiological buffer, like PBS, like phosphate buffered saline, can afford tremendous solubility for hydrophobic drugs that are permeating. And the important thing about this particular compound is that there's some evidence to demonstrate that it does not alter the permeability of the skin barrier. If you start to use other compounds, then evidence would be needed to demonstrate that that compound didn't permeate up through the skin and alter the skin barrier and violate the principles of the test. The solubility in the receptor solution is typically empirically determined in triplicate, and you want to show that you have sufficient solubility to solubilize the highest drug concentration in the IVPT pilot and pivotal study samples, ideally by an order of magnitude. You want to make sure that your receptor solution is compatible with the skin, that it doesn't alter the barrier function, that you don't have organic solvents and alcohols that may alter the skin barrier function, and that the skin is stable, and that you want to mitigate potential bacterial decomposition of the dermis and the epidermis by typically using an antimicrobial agent in the receptor solution, something like sodium azide or gentamicin sulfate. In the early days, these were not used, and that's why the length of those studies was sometimes shorter. But when using these kinds of conditions, you can often have IVPT studies that have valid conditions out for a few days. Now, the receptor solution sampling itself also needs to be qualified. How is this done? Well, the accuracy and precision of the receptor solution sample collection at each time point is qualified. And the kind of evidence that you use to qualify this typically illustrates that the sampling technique can reliably collect a consistent volume of the sample from the well-mixed volume of the receptor compartment at each sampling event. If you have a flow-through system, then the lengths of tubing and their associated dead volumes are qualified to ensure that there's a consistent lag time for the flow through the diffusion cells. And the sampling technique for vertical diffusion cells, here, for example, the sampling volume should be demonstrated that it doesn't produce artifacts like, the apparent negative flux if you're sampling aliquot maybe too low, for example. Also, you want to have available information describing the apparatus manufacturer's specification, if available, for the accuracy and precision of the sampling, especially for automated mechanical sampling systems. Now, in terms of environmental control, ambient laboratory temperature and humidity during the study is monitored and reported. Now, it's typically feasible that the temperature can be pretty precisely controlled within, for example, 21 degrees plus or minus 2 or 25 degrees plus or minus 2. The humidity can be more challenging. So it's nice to have a target, if possible, of 50% plus or minus 20%, but this is not always feasible. And if it isn't, then what's really most important here is that the test and reference products experience the same conditions throughout the test, even if that means that the humidity is changing across the duration of the test, that the test and reference products are dosed in the same donor in parallel under identical environmental conditions. For the permeation profile and the range of sampling, the flux profile and the cumulative permeation profile for the IVPT pilot study results are plotted at each sampling time across the study duration, and this profile validates that the selected study parameters have allowed you to adequately characterize the cutaneous pharmacokinetics across the range of sampling time points. So a complete flux profile is one that identifies that maximum peak flux and a decline in the flux thereafter across multiple subsequent time points in the IVPT pilot study. Now, this is not always achievable, and I think through the workshop, we can talk about what you do in those circumstances. The results of the IVPT study also validate that the sampling frequency provides a suitable resolution to adequately characterize the permeation profile, particularly the flux profile. So for example, if your first time point is your highest, is your J max, then you might not have enough resolution to be sure where your J max really lies. And you may need additional kind of more frequent sampling in that region. In terms of precision and reproducibility as part of the IVPT method validation, this is done by looking at the flux and cumulative permeation results from the IVPT pilot study again. These are calculated and tabulated and reported for each diffusion cell at each time point. The tabulated IVPT pilot data really should be clearly organized for each donor, each replicate, each treatment group, and each time point. You want to have complete results for all the data values utilized in the calculations and report them in a clear and organized manner, because you really want to be able to allow a reviewer to reconstruct those calculations using all the source data elements and to verify those calculations for cumulative permeation results or flux data. And then summary statistics are typically provided here in terms of intra-donor averages, standard deviation, and percent CV, as well as inter-donor averages and the standard error of the means, as well as the percent CV across donors. It's good to also just be aware of what the dose depletion is, but this is not requiring a large study to make it happen. This is done just based upon some simple calculations where the nominal amount of the drug dosed on the skin sections is estimated based upon the nominal strength of the drug in the topical product. So it might be, a 5% drug in product or 1% drug in product. So you know what that nominal strength is, and then you look at the approximate mass of the topical product dosed on the skin. For example, 10 milligrams of a product that's at 5% strength. Well, you know how much drug was applied on the surface of the skin. Now you compare that with the cumulative total permeation of the drug in the receptor solution across the study, and then you express this cumulative amount permeated as a fraction of the amount dosed on the skin. This is expressed as a percentage of that nominal amount, and that's essentially what you would report as the dose depletion. Typically, that's relatively small. A really important element of the IVPT method validation is discrimination, sensitivity, and selectivity. So let's talk first about IVPT sensitivity. How is this done? So what is IVPT sensitivity? It's the ability of the IVPT method to detect changes in the cutaneous pharmacokinetics of the drug as a function of differences in drug delivery. So if the bioavailability increases, the IVPT study should be able to respond and show a higher flux profile, and likewise in the other direction. The goal here is to have intentional differences that are rationally expected to produce progressive differences in bioavailability. So how do you figure out what's going to create a difference in bioavailability? Well, one way is to think about the dosage form on the skin and the metamorphosis that it undergoes. So here we have examples of metronidazole gel and cream. These are not IVPT profiles. These are drying rate profiles. So this is different amounts of the drug. And as you see, of the dosage form, this is different amounts, 3 milligrams per centimeter squared, or threefold higher, 10 milligrams per centimeter squared, or another threefold higher, 30 milligrams per centimeter squared applied of the gel, or these are amounts of the cream. And as you see, as you apply thicker and thicker doses of the product, it takes longer for that product to dry out, which means that there's more that remains on the skin for a longer duration, and therefore has a longer time duration over which that drug delivery into the skin happens. And so you can utilize these drying rate curves as a way to understand, when do I have enough of a difference in the amount dosed to the skin? This works well for those that actually have some kind of evaporative metamorphosis, that I can differentiate my drying profiles. And then when you look at these same data in an IVPT study, now looking at the flux data, you can see that these different dose amounts provide different bioavailabilities. Now, this is just meant as an illustration. You're not seeing these well differentiated because this is just one donor to illustrate the kind of data that you would get and the way that you would approach designing a sensitivity study. So the approach is to produce those differences in drug delivery that can be differentiated by a suitably discriminating IVPT method may include modulation of the dose amount, like I just showed there. This works well for products that undergo metamorphosis. But for an ointment that doesn't evaporate on the skin, this won't necessarily work. And there, you may want to actually have a modulation of the dose duration. So it's applied on for a certain amount of time, taken off, and then you evaluate the bioavailability and show that as you have different durations of the dose, that the bioavailability is differentiated by the IVPT method. Now, these differences in IVPT permeation profiles are not expected to be specifically proportional to the differences in the dose amount or the dose duration or the product strength. So for example, if you increased a product strength from 5% drug in formulation to 10% drug in formulation, you've doubled it. It's not reasonable to expect that you're going to double the bioavailability. That's not how topical products tend to work on the skin. Likewise, if you double the amount that you've applied on the skin, you won't necessarily see double the bioavailability. And so while you do want to make sure that you're not at such a high dose that the skin just can't take anymore. The skin barrier is limiting the partitioning and diffusion of the drug through, and you're at such a high dose that even if the test product had a higher bioavailability, you're already maxed out. You would not be able to differentiate that difference in bioavailability. You want to make sure that's not the level that you're at for the IVPT study. So the point of this is to be able to show that the dose that you're using is one where if there was higher bioavailability, the skin can still show and absorb more drug, and that you can differentiate that as there are differences in the amount delivered to the skin, that the IVPT profiles respond to that. Now, these IVPT sensitivity studies are typically performed, while they support method validation, they're performed during the method development phase because you're still at that point trying to establish the IVPT method parameters like the dose amount or the dose duration or the study duration. IVPT selectivity is different than sensitivity. Selectivity is different because with sensitivity, as you modulate the bioequivalence of a particular product, higher or lower, you want to see that the IVPT response is going higher or lower. Here, selectivity is the ability of the IVPT method to discriminate the cutaneous pharmacokinetics of drug between products. Or, what you want to see is differences in drug delivery from different products. So this is like, for example, between a positive and negative control for bioequivalence. So that parallel assessment in the IVPT study of the reference test and that third topical product, that third one is what shows you this IVPT selectivity relative to the reference formulation, and that provides the supportive evidence that the IVPT method is selective. And this is performed as part of the IVPT pilot study. Now, robustness, it's important to differentiate robustness from all the other validation considerations. I discuss it because it is robustness is a routine component of any typical method validation. However, one does not need to, and one may not even be able to demonstrate robustness, because here you're working with an actual human skin that exhibits biological variation. And a primary assumption related to robustness testing is that the test system performs consistently when all the system variables are at the nominal settings, like the temperature, the stirring rate. However, because of the variability inherent in the permeability of human skin, this is true in vitro or in vivo, it may not perform consistently, even when all the system variables are at nominal settings. Now, that being said, an IVPT method may be robust to certain method parameters. Like you may vary the stirring rate and say, I can have variation in my stirring rate, and I have that tolerance. It doesn't really affect my cutaneous PK profiles. So when you have results that support that your method is robust to that variation, like stirring rate, then great, report that. But ultimately, since it's not always, and what that allows you to do is then have that variation. And so, I can vary that parameter. I had variation during my study, but my study is robust to that parameter. That's the value of having that. But since there's not always, it's not always feasible to validate the robustness, the more important part is control the study procedures. Make sure that they're well controlled as precisely as possible. And then you know that the conditions that you're working with are well controlled. So how do you control it? Well, it's very important to have a quality management system. A quality management system specifically that incorporates the relevant principles of good practices for well-controlled studies. And this includes documented procedures for study personnel identification, training of personnel, qualification, understanding what their responsibilities are, study management and study management personnel responsibilities being clearly documented, having QC and QA procedures, being clear about what the responsibilities of those people are, utilizing standard operating procedures, making sure that people are trained on these, utilizing study protocols that are clearly defined and study reports that are well organized, maintaining and controlling the study facility environment and the systems there, the qualification and calibration of instruments and computerized systems, good documentation practices, also maintaining suitable records that facilitate the reconstruction of study events. So critical. Archival of study records, and this is not an exhaustive list. These are just some of the examples of the elements that are really quite important in a quality management system to have that well-controlled study. Finally, I'll just note here a few reminders about some of the elements that are relevant to any bioequivalent study. One is, the handling and retention of product samples. And you can refer here to 21 CFR 32038 or 32063, as well as these guidances for industry as applicable regarding the considerations for the retention of study drug samples. Also, you can refer to 21 CFR 32036 for information on the requirements for the maintenance of records of bioequivalence testing. This is for in vitro studies too, as well as retention samples should be randomly selected from the drug supplies received prior to dispensing during an IVRT or IVPT study in which the test and reference products are compared, and experimental observations that have the potential to influence the interpretation of study results. It's really critical that these observations, as well as any deviations, protocol deviations or SOP deviations, should be reported. With that, I have to say that it has been really my privilege to work with a phenomenal group of people throughout my career, both within the FDA, Drs. Priyanka Ghosh, Tanaz Ramazanli, Markham Luke, Hiran Patel, Sam Haidar, Elena Rantu, Ahmed Zidan, Yang, and Dr. Dennis Bashaw, all of who have contributed to a lot of the work that you've seen that I've presented here today, as well as our many collaborators, our research collaborators on FDA-funded initiatives, and just collaborators who have been mentors throughout my own career and whom I continue to learn from, Professor Audra Stinchcombe, Mike Roberts, Michael Winbergs, Narasimha Murthy, Frank Sinner, Kevin Lee, Paul Lehman, Tom Franz, and Howard Maybach. And I must acknowledge that the funding for some of the research discussed in this presentation was supported by FDA grants as part of financial assistance awards, including several of these here. So with that, I'll say thank you so much and look forward very much to the rest of this workshop.